आइए हम अनुराग जी से समझने की कोशिश करते हैं कि आपने आयुर्वेद की इस दवाई को वैशाखिक फिलॉसफी यानी कि मॉलिक्यूलर लेवल पे, वैशाखिक मीन्स एट द लेवल ऑफ फिजिक्स, एट द लेवल ऑफ एटम, एट द लेवल ऑफ साइटोकाइन, एट द लेवल ऑफ हर चीज दैट इज वैशाखिक, एंड दैट टू एट द लेवल ऑफ अलोंग विद अनुमान, कि आपने एक हाइपोथेसिस ली। तो 3 हाइपोथेसिस के बारे में मुझे आपसे समझना है कि आपने क्या किया। नंबर 1 कि आपने बोला कटु एंड कसेला, आपने 3 प्रोडक्ट ले लिए कटु एंड कसेला के। नंबर 1 आपने लिया कि इससे एंटीवायरल इफेक्ट होता है कि नहीं होता। अगर एंटीवायरल इफेक्ट होता है तो कहां पर होता है? वायरल के इफेक्ट में या तो एंट्री पे होगा, या प्रूफ रीडिंग पे होगा, या रेप्लिकेशन पे होगा। दैट्स वन। दूसरा, मेरे शरीर में क्या ये मेरी मल अग्नि को या मल पित्त को कम करता है कि नहीं करता, यानी कि साइटोकाइंस, यानी कि सीआरपी, इंटरल्यूकिन 6, डी डाइमर कम करता है कि नहीं करता, करता है तो कितनी देर में करता है। और तीसरा, अगर ये मेरे शरीर में इन्फ्लेमेशन कम करता है और सीआरपी कम करता है, तो उसके कारण से आगे जाके, जैसे ऑल इंडिया इंस्टीट्यूट में जो आयुर्वेद की मैंने जब डॉक्टर उनको बुलाया था, डॉक्टर तनुजा को, तो उन्होंने कहा कि जब हमने गिलोय और ये हमने आयुर्वेद फॉर्मुलेशन दिए, तो हमारे को पोस्ट कोविड इफेक्ट्स कम मिले। हालांकि आयुर्वेद मंत्रालय ने तो इसके अंदर में कटु और उसके साथ में मीठा भी डाल दिया है, च्यवनप्राश बोला है कि उसके लिए लो, तो उसके साथ में, क्योंकि उन्होंने कड़वा कसाला के साथ में मीठा भी डाल दिया है उसके साथ में। सो मेरे को ये बताइए, इन 3 चीजों में आप लोगों ने क्या स्टडी करी, किस लैब में करी, क्या उसका एविडेंस था, कहाँ कहाँ छपी, और उनकी ऑथेंटिसिटी क्या थी? ठीक है, जैसे आचार्य जी ने कहा, आपके पास BSL-2 लैब है, आपके पास BSL-3 लैब की फैसिलिटीज है, इसका मतलब है आप एनिमल स्टडीज भी कर रहे हो। सो अगर ऐसा है तो इसका मतलब है कि आप ACE2 रिसेप्टर्स को भी बाहर निकाल सकते हो। इसका मतलब है कि आप कोरोना के वायरस के जो स्पाइक प्रोटीन को आर्टिफिशियली, जैसे लोग आज की डेट में टेस्ट कर रहे हैं, आप उसको लैब में मिला के एंटीबॉडी न्यूट्रलाइजेशन भी देख सकते हो। तो इसके बारे में अगर ऐसा कोई एविडेंस है तो बताइए।
प्रणाम सर। सर आपके इन ज्वलंत प्रश्नों का जवाब देने में मुझे बहुत खुशी होगी, और आचार्य जी को प्रणाम करते हुए और स्वामी जी की सूक्ष्म उपस्थिति को प्रणाम करते हुए मैं आपसे अनुरोध करूंगा कि जो प्रेजेंटेशन हमने शेयर किया है, अगर हम वो प्ले कर पाएं तो मुझे आपको समझाने में थोड़ी सी सहूलियत हो जाएगी। और मैं सीधा अनुरोध करूंगा कि हम स्लाइड नंबर 2 पे आ जाएं। तो पूरे प्रेजेंटेशन में लगभग 26 स्लाइडें हैं सर, और मैं पहली 3 स्लाइड्स उन 3 मेन चीज़ों पे लेके आया हूं जो आपको और सब जनता को बताना चाहता हूं कि ये जो आप सामने देख रहे हैं, ये COPP WHO GMP सर्टिफिकेट है जो कोरोनिल को दिया गया है बाय ड्रग कंट्रोलर जनरल ऑफ इंडिया, which gives it the eligibility to export Coronil to 158 countries across the world। और जो आप वर्ल्ड का मैप सीधे हाथ पे देख रहे हैं और जहां पे वो जियो मार्क्स लगे हुए हैं, ये वो कंट्रीज हैं जहां पे कोरोनिल को एक्सपोर्ट किया जा सकता है ऑन द बेसिस ऑफ दिस सर्टिफिकेट गिवन बाय द DGCI गवर्नमेंट ऑफ इंडिया फॉर WHO GMP COPP सर्टिफिकेट ऑफ फार्मास्यूटिकल प्रोडक्ट। नेक्स्ट स्लाइड प्लीज। ये जो आप स्लाइड देख रहे हैं, इसमें मैन्युफैक्चरिंग लाइसेंस है जो स्टेट लाइसेंसिंग अथॉरिटी उत्तराखंड द्वारा दिया गया है कोरोनिल के लिए, एंड जिसमें क्लियरली ये मेंशन है कि कोरोनिल कैन बी टेकन एज ए मेडिसिन एज अ सपोर्टिंग मेजर फॉर कोविड-19। नेक्स्ट स्लाइड प्लीज। और ये WHO का जो इंटरनेशनल क्लिनिकल ट्रायल रजिस्ट्री प्लेटफॉर्म है, ये उसका स्क्रीनशॉट है जहां पे जो हमने क्लिनिकल ट्रायल कंडक्ट किया था उसकी एंड लिस्टिंग दिखाई जा रही है। नेक्स्ट स्लाइड प्लीज। तो इन तीनों स्लाइड्स को मेरा दिखाने का औचित्य और उद्देश्य सिर्फ इतना ही है सर कि जो मेन मेन टेक होम मैसेज हैं इस प्रेजेंटेशन से और डिस्कशन से, वो सबसे पहले दिखा दिए गए हैं। और अब मैं उन तरफ बढ़ता हूं जहां आपने क्वेश्चंस पूछे मुझसे, और वो बहुत ही वैलिड क्वेश्चंस है और मैं उन सबको कवर करने का ट्राई करूंगा। अभी ये जो आप स्लाइड देख रहे हैं स्टेज पे, ये बेसिक इंट्रोडक्टरी स्लाइड है कि कोविड-19 कैसे होता है। आई एम श्योर आपने अपने पिछले 1000 से ज्यादा शोज में इस सबको पूरी ऑडियंस को समझा दिया होगा कि ये कैसे काम करता है, बीमारी कैसे होती है, अब तक कितने प्रॉब्लम्स हो चुके हैं, इसके साइको सोशल बर्डन पे कितना इफेक्ट है और इकोनॉमिक्स पे भी कितना फर्क पड़ता है। इन सब बर्डन्स के बाद पतंजलि ने जो एक अप्रोच अपनाया और देखा कि इस बीमारी से कैसे कॉम्बैट किया जाए, और उसका एक तरीका है कोरोनिल, स्वसारी एंड अणु तेल, जो मैं अगले 20 स्लाइड्स में आपको कवर करूंगा। नेक्स्ट स्लाइड प्लीज। So ये कंटेंट है सब स्लाइड्स के। द बेसिक फंक्शन इज टू टॉक अबाउट द साइंटिफिक रैशनल फॉर द जेनेसिस ऑफ कोरोनिल, इन विट्रो वैलिडेशन ऑफ कोरोनिल इफेक्ट ऑन द SARS-CoV-2 स्पाइक प्रोटीन, जो सर आपने मुझसे भी कहा था उसके बारे में। Chemical composition analysis of coronil, which is a very very important one to actually show that the compound does exist in the medicine and it is there in all the batches which have been manufactured here. And then we went for evaluation of preclinical efficacy of coronil, clinical investigation, which has three main components. First, that you talked about, Anuman, what you called a randomized placebo-controlled clinical trial, clinical observation studies, and psychosomatic studies on the people who have been detected COVID positive and then who chose to take coronal succinate, and see how the psychologically the fear, the anxiety was modulated when the drug was available to them for treating themselves. Next slide, please. So here is a slide with what Pooch Acharji talked about, that we have taken close to 1,500 different plants, 1,500 different compounds from close to 200 herbal plants which are classically described in Ayurveda, and we could screen down three different plants. One is Ashwagandha, Withania somnifera, second is Giloy, Tinospora cardifolia, and third is Tulsi, Ocimum sanctum. And what we found that not all the compound from these plants were affecting the COVID virus. And when I said the effect, I simply mean to say that the way virus could enter inside a human host cell, or it could get replicated inside the host cell. So first two things what you see on the top of the slide, Withinone and tinocorticide, they were actually detected or predicted to inhibit the entrance of the virus inside the host cell. And that entrance is actually done by the spike protein's receptor binding domain, binding it to the ACE2 receptor of the human host cell. And we could find that the Withinone and tinocorticide could inhibit that interaction at a much higher significant binding energy reduction. We could do the simulation and could find that yes, even for close to 1000 nanosecond simulation, those interactions were broken up. The salt which were made between these two proteins, those were also broken up. Sculptrine, which is a compound from Tulsi, this actually binds very well to RDRP. RDRP stands for this RNA-dependent RNA polymerase, which is a specific protein required for the virus to make a copy of its RNA. As most of your audience would be knowing it, that SARS-CoV-2 is a RNA virus, so that making a multiple copy of the virus is actually very important, which get inhibited by the compound coming from Tulsi. On the same slide, I have also already marked. Can I have the slides? Yeah. So the work that we have done for Withinone and tinocorticide has been already published. The work for Withinone is right now in press for drug design, discovery, and development, which is a journal from Dow Press, impact factor close to 4. And the study from tinocorticide have been also published in the Ventum publication of computational modeling. What I also want to say that once we published or put up a preprint of Withinone in March 2020 in Springer Nature preprint server, it excited other researchers in India too. And at the bottom of the slide, you could see the third-party validation, what we say, that people also start to look for different components of ashwagandha, whether they can have an efficacy or at least a computational efficacy in the interaction of virus with the host cell. And we are very happy to say that our ignition towards the thought came out as a pretty good success from the other group too. Next slide, please. So this is a biochemical validation. So what we saw earlier was a computational prediction, but does it really happen inside a cell, or does it really happen when you have two binding proteins present? So this is the molecular interaction that you asked about, sir, whether two molecules really interacts. And the answer is yes. We actually procured the spike protein from SARS-CoV-2 and also got the ACE2 protein, the one they talked about, which is present in the human cells, and could actually have them interact inside the test tube in a cell-free system. That's we call biochemistry. And then we measure the interaction of that and in presence of the Withinone and tinocorticide. and we could find that Vidinone indeed inhibited that interaction in a concentration dependent manner with a very, very specific IC50, what you generally see for a pharma industry kind of compound. The IC50 was found as 0.33 nanogram per ml, which is actually pretty much strong as you expect for a compound to show that kind of efficacy. That work has been also published in the same journal that we talked about. So the next slide, please. So this one talks about the further biochemical validations of these interactions, and then we chose to do those works in by the Coronil as well. So the point there is that Coronil was formulated by using ashwagandha, giloy, and tulsi, and the purified compound from ashwagandha showed an efficacy. Then we went ahead and thought, what does happen if we take Coronil as such and try to look for those parameters? Next slide, please. So this slide actually shows the interaction between ACE2 with the SARS-CoV-2 spike protein, whether that is getting inhibited by the Coronil. So the left graph on the top side shows the concentration dependent change in the interaction parameters, and this is done with the wild type, or at that time whatever was the wild type of the spike protein present there. And then from the instructions of Shri, we went ahead to look for the different mutants which were prevalent at that time when we did the research. And we are happy to report that we looked for two different mutations, not the one that we talked today. We looked at the D614G and W436R. So these are the two strong mutants which have been reported for COVID virus. So we got a spike protein from that mutation and could actually show that Coronil indeed inhibits those mutant spike protein interaction with the ACE2 as well, which would actually indicate, yes, indeed, if there is an mutant form available, or if a person having these kind of infection with these mutant viruses, mutant COVID viruses, Coronil would be able to take care of that as well. Next slide, please. So here we come to the next part. What we talked about, sir, about cytokines. Cytokines are the marker, and as you said very correctly, cytokines are indeed the master player, the molecular switches that control inflammation. So we chose to look for this approach where we took the human lung cell lines A549 and took these three different spike proteins that we've shown in the earlier slide, and we dumped those spike protein on A549 cells and looked for the response for three major cytokines: IL-6, Tnf alpha, and IL-1 beta. And what really happens that these spike proteins does induce the cytokine release. That means the cytokines were there in the cell soup. We could measure that by using ELISA, and in presence of Coronil, it was also inhibited in a concentration dependent manner. So that actually went ahead to show and gave us a very clear indication Coronil indeed inhibits the excited level of cytokine secretion which happened by the viral proteins. Next slide, please. These studies are also published in the Journal of Inflammation Research, Impact Factor 5. And as we saw earlier, we also went to go for the next stage. And this is one of the classic experiments, and I'm very happy for my microbiology team led by Dr. Swati Haldar with other team members, Dr. Hushar Singh as well, where we went ahead and looked for a scientific collaboration from Germany, Professor Stephen Pohlmann from Linzwig Institute. He was very kind to share the expertise he has developed, and the expertise was to generate pseudovirus. Since we could not culture a SARS-CoV-2 virus because it's infective in nature and required BSL-3 facility, even up to a level of BSL-4, we had a BSL-2 facility. So we chose to manufacture a virus which would have a spike protein of SARS-CoV-2 on the surface of it, but it would be non-infective in nature to deal with. And smartly, we also filled up a luciferase gene, the gene that's going to light up whenever the virus is present inside that virus. So that pseudo virus was assembled, manufactured in the research laboratory of Patanjali Research Institute. As you correctly said, that's the one thing one should do. And we're very happy to report and feel very proud about it that we did it 6 months before. and generated those virus and then look for the entry of those pseudovirus inside the human lung cells. We got two different cell lines. One is A549 cells, which is the lung alveolar cells, and we also got BS2B cells, which is the upper airway cells coming from the bronchus. In A549 cells, we could clearly see, if you look at the slide at the bottom right, where we have the word says entry inhibition, the bar chart shows very clearly that indeed coronil could inhibit the viral entry inside the human cells. And that was the proof of concept we are looking for. This was the first cell biological proof of efficacy that we could see that coronil inhibited the viral entry in a concentration dependent manner. The pink bar that shows it starting from 1 microgram per ml goes up to the 10 microgram per ml. And not only coronil inhibited the viral entry, it also inhibited the pseudovirus induced cytokine release from these cells. And the bar chart in the center that you see around an oval shape shows again the same three cytokines, IL-6, Tnf alpha, and IL-1 beta. All three of them got inhibited in a dose-dependent manner by coronil. So that shows the coronil treatment could rescue the cells, in this case, from the cytokine storm. This work has been, of course, published in the Journal of Inflammation Research. Next slide, please. So here comes the chemistry. So once we have a drug-like molecule or a medicine in our hand, it's empirical and it's very important that we show what are the chemical constituents for it. And we actually employed three different technologies to actually confirm that. The first one, what you're seeing on your screen, is the LCMS, liquid chromatography mass spectroscopy, where we could actually identify 44 phytocompounds present in coronil tablet. And we did for multiple batches, multiple tablets, and we could actually get pretty much the similar data every time. Next slide, please. Here, what you're seeing is Hplc, high performance liquid chromatography, where we could actually quantify the amount of compound presented over there. So we could see close to 11 different compounds that we could identify from different batches, and which again have been now published in Journal of Inflammation Research. Next slide, please. And this slide shows you HPTLC, high performance thin liquid chromatography, where we could actually look for the specific compounds and could confirm whatever the finding we had from HPLC. So what you see, the gels on the top, which are there for the purified compound, the normal standards, and the band in the center talks about the coronils. So that shows that yes, those compounds are present in the right amount, and it is there from all the batches. We have now data for 50 odd batches, and our recent publication, which is out there very soon, are talking about the 15 different batches of coronil tested across a 3 months time duration of manufacturing. Next slide, please. So once we finish the in vitro work, then the challenge happens that what we can do in vivo. And I'm sure, as most of you would be knowing it, COVID virus does not infect rats and mice. The rodents are actually not sensitive for COVID virus. So that becomes very challenging, because most of the time when you do the experiment with the animals, you go towards mice and rats. So then the thought comes that what we can do, is there something that we can explore? And then we chose to go towards zebrafish. So zebrafish is an vertebrate and is a very well established animal model for variety of research. And we have done considerable amount of work in past, including the work on dengue, which is the medicine for dengue, which was done earlier, and we could show that we could infect zebrafish with the dengue virus and could actually rescue the effect of it. So we chose, if we already had a past success, how would we do it now? But the trouble happens, the COVID virus is a virus of respiratory system. It affects the lungs at the first place. And the fish, unfortunately, have no lungs. So how are we going to do about it? So then we chose to look for the analogous organ inside the fish. And we found swim bladder is an air sac which is there inside the fish, which is used by the fish for remaining buoyant. It used to take it going up and down inside the water. So we then went ahead, took the human lung cells, transplanted inside those air sac, and waited for several days for those cells to get stick over there and grow. Once that happened, we get a humanized zebrafish model, and those humanized zebrafish were then challenged with the spike protein of SARS-CoV-2 virus. And as expected, the SARS-CoV-2 spike protein induced severe form of inflammation, which could be measured by a different cytology, which you can see in these slides. And you can see the different inflammatory cells migrating like granulocyte, lymphocyte, and macrophages at the site of the injury. We could also measure the behavioral fever in the fish, and we could also follow it up the cytokine response that we see inside an intact animal. So the next slide shows you the data. Well, I think this one slide is missing. Can we go back to earlier slide, please? Yeah, so this slide shows the different cytological parameters. This maybe very busy slide to show, but I want to take your attention towards the right hand side where you're looking the set of bar charts, 4-day and 7-day. So these are the data where you're looking for cytokines present inside the cytokines present in the serum of the fish, which get inhibited by the human relevant doses of coronil in a dose-dependent manner. And we look for the three different cytokines as usual, IL-1 beta, TNF alpha, and IL-6, and they get inhibited considerably. In addition, coronil also rescued the fish from behavioral fever. It ameliorated the inflammatory response and also prevented the kidney necrosis. These results have been published in the Journal of Molecule Impact Factor 3.5 3 months before. Next slide, please. I think we jumped a slide. There's one more slide in between. Can you go back, please? One more. Yeah, this slide. So this slide is the slide which I think going to take the attention from Dr. K.K. Aggarwal, that this slide shows the molecular pathway which might be getting inhibited in order to control the cytokine response. So we could show from our in vitro, biochemical, as well as animal experiments that coronil indeed moderated the expression level of IL-6, TNF alpha, and IL-1 beta. Then the question comes, under what pathway? So we could find, this is there out there in the literature from other work, that ashwagandha, giloy, and tulsi indeed could play at different switches of this molecular pathway. We found that IRF3, we also found for TVK1 and NF-kappa B, which get inhibited by ashwagandha and tulsi, whereas a giloy could actually moderate the expression in the pathway of P38, JNK, and also a little bit of NF-kappa B. So those transcriptional control of IL-6, TNF alpha, and IL-1 beta would be a right way of controlling these release in the cell lines, in the animals, and of course, in the humans. The data of humans I'm going to show in my next slides, please. Next slide, please. So here comes about the randomized placebo-controlled clinical trials. These trials were conducted at NIMS University, Jaipur, under the leadership of Professor B.S. Tomar. These were conducted in the month of May and June. And at that time, the corona was at its prime, and we could get hold of a COVID center where the COVID positive patients were actually recruited. So all of these patients in this trial were COVID-19 positive by RT-PCR. Close to 100 patients were recruited, and they were divided into two groups. One group was giving a placebo, which is one of the rare thing in Ayurvedic drugs. So said it very nicely that, randomized trials are very important, but having a placebo-controlled randomized trial, it's the real requirement for any success. So the team from Patanjali, the team from Divya Pharmacy were very kind enough to generate the placebo tablets, which look and feel like the normal medication we have. And here we actually had the main components of coronil, which is ashwagandha, giloy, and tulsi, along with swasari and anutela, which were given to the patients. And the primary endpoint was to look for the rate of recovery from a COVID positive getting COVID negative, and number of days it takes it. Next slide, please. So this slide is actually a summary result that you can see very easily. Primary endpoints, positive become negative. The bar chart on the left shows that the placebo group, which is orange in color, and green color bar shows the treatment group. So patients within 7 days got completely recovered. 100% recovery was observed in the patients who were given the Ayurvedic medicines, whereas the placebo group got a 60% recovery, which were actually very exciting, which was the first primary target we set up for our experiment, and our team were very happy to report that yes, indeed, it meet the target that we set it out. We could also see the uh, pr- proportion of recovery, which actually was faster in the treatment group. Next slide, please. Next slide, please. Excuse me. Can you hear us, please? Sir, are we audible to you?
आ रही है आवाज?
Sir, can you go to the next slide, please? I've just 3 more slides left. No, I think we went too fast. A few more slides earlier, where we're going to show the C-reactive proteins level.
वापस जाओ,
One more
पीछे
slide,
जाओ.
please.
यस, ये वाला.
ये वाला, ये. सो इस स्लाइड में हम ये देख पा रहे हैं जो प्लेसिबो कंट्रोल है, जो ऑरेंज कलर के बार हैं, और जो ट्रीटमेंट है जो ग्रीन कलर के बार हैं, कि फ्रॉम डे 1 टू डे 7 प्लेसिबो में सीआरपी लेवल कितना था और जो ट्रीटमेंट है उसमें सीआरपी लेवल कितना था. एंड इट्स वेरी क्लियर दैट देयर इज अ स्टैटिस्टिकली सिग्निफिकेंट change in the serum CRP level of the patients who were given Ayurvedic medicines. It was 12.4 times lesser than the placebo group. This study, the whole clinical trial outcome, which I am talking in last couple of slides and two more slides, have been published recently in the journal Phytomedicine. Impact factor 4.5. Next slide, please. Here we see the TNF alpha. So this was a cytokine that we could focus, and we could get the reagent and have that been measured in the patient serums. And as you can see, similarly like HCRP, the TNF alpha level was also significantly downregulated in the patients who were given the Ayurvedic treatments. Next slide, please. So that was the data we had for the controlled clinical trial conducted in Jaipur. In parallel, or slightly earlier to that, we also could conduct a clinical case study in some patients in the COVID center in Ahmedabad, where the people were given a choice to take Ayurvedic medicines alone or in parallel with the allopathic medicines. And there was close to 500 patients who were given that option, and they chose to do that therapy. And we are happy to report that the patients who were given the Ayurvedic medication shows a much faster recovery from COVID-19 infection. This work is under final review in Journal of Herbal Medicine. Next slide, please. And this is the one that I was talking about, the influence of patient reporting treatment satisfaction on the psychological impact and quality of life by Coronil. So these are the patients, close to 367 patients who were actually interviewed for a various parameter of the quality of life, also anxiety, stress, effectiveness, and the convenience, as well as the global satisfaction for the treatment. And the basic questionnaire design was done in such a way that they were asked whether they were having a different degree of depression, anxiety, and stress with the COVID-19, and if the drug is available in form of in form of Coronil associate, how was their level? And we could actually see very clearly that out of those 367 patients, we could see that increased treatment satisfaction with the Coronil kit lead to decreased psychological impact and increase the quality of life in all of the study participants. This work is right now under final review in Journal of Clinical Medicine. Next slide, please. And probably the last slide, yes. So summary, the scientific summary that we talked about, sir, so far is that we have observed, reported, and talked about the pharmacological effects of Coronil as an inhibitor of SARS-CoV-2 spike protein mediated entry of virus into the lung cells, that we established it beyond any reasonable doubt. Coronil disrupt the interaction between the SARS-CoV-2 spike protein and human ACE2 receptor. Coronil modulated SARS-CoV-2 spike protein induced cytokine storms in the human lung cells. Coronil ameliorated SARS-CoV-2 spike protein induced pathological symptoms in humanized model of disease, which is a zebrafish model we talked about. And I'm very happy to report as well, the data is not shown here, which is a very important point too, that we have conducted 28-day repeated tox study in rats and rabbits under GLP norms for Coronil and went up to a 1000 milligram per kg daily dose. So this is 1000 milligram per kg dose of Coronil, give it for 28 days in parallel, and in rats and in rabbits. And we are happy to report there was not a single tox finding that have been shown. So this is actually almost 10 to 15 times of the human dose that we are talking about. So this is a good tox data which is done under GLP mode. In clinical studies at NIMS University, Coronil expedited the recovery in COVID-19 positive patients. As I said, 100% recovery was observed within 7 days as compared to 60% seen in the placebo group. And in our psychosomatic studies, there are actually more than one now, where we could show that Coronil treatment exhibited a significant satisfaction leading to reduced stress, anxiety, depression, along with the improved quality of life. Thank you very much.